Students, we are discussing isolation of a desired gene. In the isolation of desired gene, we are discussing restriction enzymes. What it is? Restriction enzymes. Now, listen this statement. There are 4 types of nucleotides in DNA. Yes or no? There are 4 types of nucleotides in DNA. I am speaking about DNA. Sir, DNA contains how many types of nucleotides, sir? 4 types of nucleotides. What are they, sir? Adenosine monophosphate. Strictly speaking, it is a deoxyadenosine monophosphate, deoxyadenosine monophosphate, deoxyguanosine monophosphate, deoxyguanosine monophosphate, deoxythymidine monophosphate, deoxythymidine monophosphate, deoxycytidine monophosphate, deoxyadenosine monophosphate, deoxyguanosine monophosphate, deoxythymidine monophosphate, deoxycytidine monophosphate. These are 4 types of nucleotides present in a DNA. These are 4 types of nucleotides present in a DNA. Hence, the probability of that any one of this occur at a place is how much, sir? 1/4. Therefore, what is the probability of any one out of these 4 is what, sir? 1/4. For example, I am writing the sequence. See here, ATGC. How many type of nucleotides? 4 type of nucleotides. 4 type of nucleotides. Is it okay? Right? Again, A will come as which nucleotide, sir? 4th nucleotide. See here, sir, this is right A. Again, A is coming as which nucleotide? 4th nucleotide. 1, 2, 3, 4. Right? Sir, take T, sir. Sir, T is repeating as which nucleotide? 4th nucleotide. 1, 2, 3, 4. Sir, take G, sir. Coming as which nucleotide? 4th nucleotide. 1, 2, 3, 4. Yes or no? 1, 2, 3, 4. Is this statement okay? Right? Therefore, what is the probability of each nucleotide? What is the probability of each nucleotide? 1/4. Sir, why we are taking 1/4, sir? Because DNA is having only 4 type of nucleotides. In that 4 type of nucleotides, the probability of any nucleotide will be how much? 1/4. Is this statement okay? Right. Therefore, there are 4 types of nucleotides in a DNA. Hence, the probability of any of this nucleotide, right? The probability of that any one of this occurs at a place is how much, sir? 1/4. Is it okay? Right. Now, see this statement. Assuming that the occurrence of bases in adjacent position, occurrence of what? Bases in adjacent position, right, is random. Then what is the probability that GAA-TTC sequence occurs? Probability of GAA-TTC sequence occurs. How much, sir? 1/4 to the power of 6. How much it is? 1/4 to the power of 6. Sir, 1/4 to the power of 6 means what, sir? It will be 4096 nucleotides. It means for every 4906, for every 4096 nucleotides, this sequence will repeat. What is that sequence? GAA-TTC sequence. Is this statement okay? Right? Therefore, what is the probability of GAA-TTC sequence, sir? 1/4 to the power of 6. That is how much? 4096 nucleotides. 4096 nucleotides. It means for every 4096 nucleotides, this GAA-TTC sequence will repeat. Is this statement okay? Sir, how we got this 1/4 to the power of 6, sir? Now listen here. What is the probability of each nucleotide in a DNA? Probability of each nucleotide is how much? 1/4. Is it okay? Right. Sir, GAATTC. This sequence contains right how many nucleotides. I am asking number of nucleotides, I am not asking type of nucleotides. In this sequence, how many nucleotides are there, sir? In this sequence, 1, 2, 3, 4, 5, 6. 6 nucleotides are there. Is it okay? How many nucleotides are there in this sequence, sir? 6 nucleotides are there. Now you tell me, in DNA, what is the probability of each type of nucleotide? Probability of each type of nucleotide is 1 by 4. In this sequence, how many nucleotides are there? 6. Therefore, 1 by 4 to the power of 6. It becomes how much? 4096 nucleotides. Therefore, this sequence repeats for every 4096 nucleotides. Is this statement okay? For example, sir, the sequence is GAAT, sir. This is a sequence, sir. This sequence, right, this sequence, what is the probability of this sequence, sir? How we will calculate? See here. What is the probability of each type of nucleotide in DNA? Probability of each type of nucleotides in DNA, 1 by 4. In this sequence, how many nucleotides are there? 4 nucleotides. Therefore, 1 by 4 to the power of 4. 1 by 4 to the power of 4. 1 by 4 to the power of 4. Sir, for example, the given sequence is GAA, sir. What is the probability of occurrence of this sequence, sir? Probability of occurrence of this sequence. Probability of occurrence of this sequence, sir. What is the probability of each nucleotide in DNA? 1 by 4. Probability of each nucleotide in DNA is 1 by 4. Sir, in this sequence, how many nucleotides are there? 3 nucleotides are there. Therefore, 1 by 4 to the power of 3. 1 by 4 to the power of 3. Therefore, is this statement clear? Is it okay? Right? Therefore, GAATTC sequence repeats for every 4096 nucleotides. Is it okay? Clear? Right? Listen here. Sir, this GAATTC sequence occurs for every, right, 4096 nucleotides. Is it okay? Right? However, in reality, the location and frequency of such sequences GAATTC, right, in a given genome, is it random? It's not random, but it is a species specific. Is this statement okay? Sir, we are saying that GAATTC sequence repeats for every 4096 nucleotides. 4096 nucleotides. Yes or no? Right? But in reality, the location and frequency of such sequences, in a given genome is not random. It's not random, but it's a species specific. It's species specific. Is this statement okay? Right? Now listen this statement. When DNA cut by same restriction enzyme, when DNA cut by same restriction enzyme, right, we will get same sticky ends. What we will get, sir? Same sticky ends. When we will get same sticky ends, sir? If we cut the DNA by the same restriction enzyme, we will get the same sticky ends, sir. So, such DNA fragments can be joined. Such DNA fragments can be joined by what, sir? DNA ligase enzyme. What are joined? DNA fragments are joined. How DNA fragments are joined, sir? One end of the fragment is joined to another end of the fragment. Therefore, they are joined end to end. Joining is also called as ligation. Joining is also called as what? Ligation. Therefore, we are calling this technique as end to end ligation. What is this technique called as, sir? End to end ligation. One end of the fragment is joined to another end of the fragment by using DNA ligase enzyme. So we are calling this technique as what? End-to-end ligation technique. End-to-end ligation technique. Is this statement okay? Right. Now listen this statement. Sir, by using restriction enzymes, we are cutting the DNA molecule. Cutting the DNA molecule. This is called as restriction enzyme digestion. What it is called as? Restriction enzyme digestion. Right. Therefore, cutting what? Cutting DNA by what, sir? Restriction enzymes. Cutting DNA by restriction enzymes. What is this technique called as a? This is called as a restriction enzyme digestion. What it is called as? Restriction enzyme digestion. Restriction enzyme digestion. Is this statement okay? Right? Now listen here. Sir, when we cut the DNA by restriction enzymes, do we get only one fragment? No, we will get many fragments. We are getting many fragments. We are getting many fragments. Is this statement okay? Right? Sir, do all the fragments are of same length? No. They are of different length. They are of different length. Is this statement okay? Right? Listen this statement. When we cut what? DNA by restriction enzymes, we get what? DNA fragments. What we will get, sir? DNA fragments of same length or different lengths? Of different length. Sir, take one DNA molecule and cut that DNA molecule by using a restriction enzyme. Then what happens? You will get many fragments. Do length of all that fragments is same? No. You are getting fragments of different length. Is this statement clear? Is it okay? Right? We are discussing restriction enzymes, right? Now go for nomenclature of restriction enzymes. Nomenclature of what's a restriction enzymes? Nomenclature of restriction enzymes. It means how these restriction enzymes are named. Listen here. Sir, in a restriction what? Enzyme. In a restriction enzyme, right? First letter. What's that? First letter stands for genus of bacteria, from where it is isolated from. Whereas second and third letters, what and what, sir? Second and third letter stands for species of bacteria, from where it is isolated from. Whereas fourth letter, what's sir? Fourth letter, it stands for strain. It stands for what, sir? Strain. Whereas Roman number, what? Roman number, it indicates what? Order of discovery, order of isolation. What, sir? Order of isolation. See here, sir, first letter indicates genus of bacteria. Second and third letter indicates what, sir? Species of bacteria. Whereas the fourth letter indicates a strain. Whereas Roman number indicates what, sir? Order of isolation. Roman number indicates what, sir? Order of isolation. Order of isolation. Is it okay? For example, take a restriction endonuclease enzyme. Restriction endonuclease enzyme by name EcoR1. What is the enzyme name? EcoR1. Sir, what is the first letter? E. Right. Sir, first letter, what it is? First letter, it indicates what, sir? Genus. What is a genus? From where this enzyme is isolated from? Escherichia. Is it okay? Sir, second letter and third letter. What and what, sir? Second and third letters. Second and third letters represents what, sir? Second letter and third letters represents a species. What is the species name, sir? Coli. Whereas the fourth letter, what is the fourth letter? Sir, fourth letter represents strain. Fourth letter represents what, sir? Strain. What is the strain here, sir? The strain is a RY13 strain. RY13 strain. RY13 strain. Whereas Roman number, what, sir? Roman number. Here, what is the Roman number? Roman 1. What it indicates? Order of isolation. Order of isolation. It means from this strain, which strain? RY13 strain of E. coli bacteria. RY13 strain of E. coli bacteria. This is the first isolated enzyme. So it was indicated with Roman 1. I am not saying that this is the first discovered restriction endonuclease enzyme. Is it okay? Right. From E. coli RY13 strain of bacteria. From E. coli RY13 strain of bacteria, right, this was the first isolated enzyme. What is that? EcoR1. That's why Roman number 1 is given. Is it okay? Clear this statement, right? Therefore, sir, here what is the first letter, sir? First letter indicates genus. What is the first letter actually in this enzyme name? E. Sir, second letter and third letter. Here, what is the second letter, sir? C. What is the third letter, sir? O. Therefore, C, O. Second letter, third letter. Indicates what, sir? Species. Fourth letter. What is that fourth letter here? R. R is the fourth letter. Right. What it indicates? Strain. What is the strain here? RY13. What is the Roman number here? 1. Roman number indicates order of isolation. It means, right, from E. coli RY13 strain of bacteria, this is the first isolated enzyme. Is it okay? Therefore, EcoR1 is isolated from which bacteria? E. coli RY13 strain of bacteria. Is this example okay? Now go for second example. Go for second example. Hind 2. Take a Hind 2. Hind 2. Now see here. Sir, here what is the first letter, sir? H. H should represent what? Genus of bacteria. What is the genus, sir? Haemophilus or Haemophilus. Sir, second letter and third letter, sir, indicates species. Influenza. What it is? Influenza. Okay, right. Whereas the fourth letter, what is the fourth letter? D. What it indicates, sir? Strain. D strain. Sir, what is the Roman number, sir? Roman 2. What does it indicate? What does it mean? Roman 2, what it indicates? This is the second restriction endonuclease enzyme that was isolated from D strain of Haemophilus influenza bacteria. This is the second restriction endonuclease enzyme. What it is? Hind 2. Right. Therefore, this is a second, what, sir? This is the second what? Restriction endonuclease enzyme that was isolated from D strain of Haemophilus influenza. Is this statements okay? Clear. Therefore, Roman 2 indicates order of isolation. Order of isolation. Clear, right? Listen to nomenclature of what, sir? Restriction enzymes. Now listen here. Now I am continuing the flow chart. What we drawn, right, in the first class. Okay, of isolation of desired gene. Isolation of desired gene. Okay, now listen here. Sir, by spooling process we isolated the DNA which is in pure form. Yes or no? Right. Now this DNA which was isolated, we are cutting the DNA molecule by using restriction endonuclease enzymes. Till now we discussed this restriction endonuclease enzymes. When we cut the DNA by using restriction endonuclease enzyme, we got fragments of DNA. What we got? Fragments of DNA. All those fragments are not of equal length. Yes or no? We got what, sir? Fragments of DNA. Now what we have to do means we have to separate these fragments of DNA from one another. Separation of DNA fragments. How you are going to separate these DNA fragments, sir? DNA fragments are separated by using gel electrophoresis. By using what, sir? Gel electrophoresis. Which gel electrophoresis, sir? Agarose gel electrophoresis. By using agarose gel electrophoresis, these fragments of DNA are separated from one another. Therefore, what are separated? DNA fragments are separated. Therefore, separation of DNA fragments. Sir, by which technique these DNA fragments are separated from one another, sir? By agarose gel electrophoresis. By agarose gel electrophoresis. Therefore, now, right, I am keeping this flow chart aside and now we are going to discuss this agarose gel electrophoresis. What does this agarose gel electrophoresis stands for? Is it okay? Right. First, what is the meaning of this agarose? Sir, agarose is a polysaccharide. Agarose is a polysaccharide. Sir, polysaccharide means it should be a polymer. It should be a polymer of what, sir? Sugar. It should be a polymer of what, sir? Sugar. Sir, this agarose is extracted from. Agarose is extracted from where? Extracted from marine seaweeds. Extracted from where, sir? Marine seaweeds. Marine seaweeds. Sir, from which marine seaweeds it is extracted, sir? Red algae. Red algae means comes under which class, sir? Rhodophyceae. Rhodophyceae. Red algae means comes under class Rhodophyceae. Is it okay? Therefore, agarose, it is a polysaccharide. It is a polymer of sugars extracted from marine seaweeds. Which seaweed, sir? Red algae. Red algae means comes under class Rhodophyceae. Is it okay? Sir, agarose, as it is extracted from marine seaweeds, therefore agarose becomes a what, sir? It is a natural polymer. It is which polymer, sir? It is a natural polymer. It is which polymer? Natural polymer. Is this statement okay? Right. Now go for electrophoresis. What is the meaning of electrophoresis, sir? See, electro means electricity. Phoresis means movement. It means a molecule is moving in response to electricity. In response to what? Electricity. Therefore, movement of a molecule or separation of a molecule in electric field is called as what, sir? Electrophoresis. What it is called as? Electrophoresis. Okay, right? Electro. Energy coming from where? Energy from electricity or electric field. Is it okay? Right? Next, phoresis. What is the meaning of phoresis? Movement. Phoresis means what, sir? Movement, separation. Therefore, sir, electrophoresis, separation or movement of what? Charged molecules. Charged molecules. Separation of charged molecules in where? Electric field. Separation of charged particles or charged molecules in electric field. This is called as a what, sir? Electrophoresis. What it is called as a electrophoresis. Sir, which charged molecules are separated by electrophoresis, sir? Which charged particles or charged molecules are separated by electrophoresis, sir? nucleic acids. Nucleic acids. Nucleic acids means what and what will be? DNA and RNA. Sir, nucleic acids are made up of what? Nucleotides. Therefore, we can separate nucleotides also. Next one is proteins. What's sir? Proteins. Nothing but polypeptides. Polypeptides made up of what? Amino acids. Therefore, even amino acids. Therefore, what are the charged molecules that can be separated by electrophoresis? Nucleic acids, that is DNA and RNA, nucleotides, proteins, polypeptides, amino acids. These are the charged molecules that can be separated by electrophoresis. Is this statement clear? Okay, right? Right, agarose gel electrophoresis, right? Now this is the kit, KIT, kit. We are not speaking about KCR kit, right? This is the kit in which a gel electrophoresis takes place. Is it okay? Right. Sir, in this kit, two electrodes are present, cathode, right? Negative electrode. See, this is a cathode. Which electrodes are? It is a negative electrode. Whereas this is a anode, right? Sir, anode. Which electrodes are? Positive electrode. Is it okay? Right. Now, sir, what are these white boxes, sir? These white boxes are nothing but wells. What are these white boxes, sir? Wells. All these white boxes are wells. In these wells, we are taking DNA fragments. What we are taking, sir? We are taking DNA fragments into these wells. Is this statement okay? Clear? Right. Now, see here. Sir, here how many wells are there in this diagram? 4 wells, right? Sir, I am numbering these wells. See, sir, this is the first well, this is the second one, this is the third one, this is the fourth one. Sir, in first well, in which well, sir? First well, I am calling this one as first lane. First well, first lane. I am taking, right, I am taking undigested. What? Undigested gene. Nothing but standard. Nothing but what, sir? Standard. Whereas in a what, sir? Second, third and fourth wells or lanes, I am taking digested DNA fragments. Is this statement okay? Clear? Right. Therefore, sir, when we treat a DNA by restriction endonuclease enzymes, we are getting fragments of DNA. Yes or no? We are getting what? Fragments of DNA. Is it okay? Right. I am taking that fragments of DNA in second, third and fourth lanes. Is it okay? Sir, in these fragments of DNA, one will be the desired gene. We know that gene. It means what is the gene we are interested in? That gene we know. Yes or no? Therefore, out of these fragments of DNA, one will be the which genes are? Desired gene. One will be the desired gene. Okay. Is that statement clear? What gene we want to isolate? We are taking that gene in the first well, that is the first lane. Is this statement okay? Right. Now, I am filling this kit with what's our agarose gel. This total kit is filled with which gels are? Agarose gel. Filled with agarose gel. Is this statement okay? Right? Filled with which gel? Agarose gel. Now you supply electricity. What is supplied? Electricity is supplied. When electricity is supplied, see, in these wells, what are present? DNA molecules. In these wells, what are present, sir? DNA fragments are present. In these wells, what are present? DNA fragments are present. DNA is negatively charged. Sir, negatively charged DNA fragments, they move towards anode, that is positively charged electrode. Yes or no? It means now the DNA fragments, they are moving through Which gel? Agarose gel by sieving effect. By which effect, sir? Sieving effect. By which effect? Sieving effect. Is this statements okay? Right? Therefore, what are moving? Negatively charged DNA fragments. Which fragments, sir? Negatively charged DNA fragments. Negatively charged DNA fragments moves towards anode through agarose gel by sieving effect. By which effect? Sieving effect, and gets separated according to their size. It means separation is according to their size. Separation is according to their size. How you are saying that separation is according to their size, sir? See here. For example, here how many DNA fragments are there? 4 DNA fragments. I am assuming that out of 4 DNA fragments, this is of 8 centimeters in length. This is only our assumption. Sir, 6 centimeters length, right, 4 centimeters length, right, 2 centimeters length. See here. Sir, this is the smallest fragment out of these 4. Its movement is very fast. That's why it came near to anode. Sir, this is the largest fragment. This is which fragment, sir? Largest fragment out of these 4. Right, its movement is slow. Its movement is slow. That's why it is near to the well. Sir, all these fragments were taken in these wells, yes or no? When we supplied electricity, the fragments present in this well started moving in this agarose gel. Is it okay? Clear? Right? And these fragments, they get separated according to their size. Is it okay? Smallest fragment is near the anode, whereas the largest fragment is near to the wells. Therefore, separation of DNA fragments has taken place according to their size. Separation of DNA fragments taken place according to their size. Is these statements okay? Right? Therefore, now listen these statements. Listen these statements. I am speaking about agarose gel electrophoresis. Right? Negatively charged what? DNA fragments. Negatively charged DNA fragments moves what? Moves towards anode through what? Through agarose gel. Through agarose gel, what are moving? Negatively charged DNA fragments are moving towards what? Anode by sieving effect. By which effect, sir? Sieving effect, and gets separated. What are separated? DNA fragments are separated and get separated according to their size. It means separation is according to what, sir? Separation is according to the size. Is this statement okay? Clear? Right. Now listen this statement. Sir, smallest fragment. Sir, where smallest fragment is, sir? Smallest fragment, right, near to what? Near to anode. Near to what, sir? Anode. Away from well or away from cathode. Is it okay? Right? Sir, where is the largest fragment, sir? I am speaking about which fragment, sir? Largest fragment. Largest fragment. Sir, where is the largest fragment, sir? Near to cathode or near to well, whereas away from what? It is away from anode. Sir, why smallest fragment is near to anode? Why largest fragment is near to cathode, sir? Listen this statement. Smaller the size of the fragment, faster is its movement. Smaller the size of the fragment, faster is its movement. Therefore, smallest fragment moves fastly, whereas the largest fragment moves slowly. Is this statement okay? Right? Or what? Smaller the what? Smaller the size of a fragment. Smaller the size of a fragment, faster is its movement. Faster is its movement. Smaller the size, faster is its movement. Is this statement okay? Right? Therefore, smallest fragment moves fastly. Largest fragment, how it moves? Slowly. Largest fragment moves slowly. Is this statements okay? Right. Therefore, smallest fragment moves fastly, whereas the largest fragment moves slowly. Is this statements clear? Okay. Right. Sir, in agarose gel electrophoresis, in what, sir? Agarose gel electrophoresis, separation of DNA is based on what? Separation of DNA is based on size. Separation of DNA is based on size. Separation of DNA is based on size. In agarose gel, what? Electrophoresis. In agarose gel electrophoresis, separation of DNA fragments. Separation of DNA fragments is according to what? According to size. Separation of which fragments? DNA fragments. Is according to what, sir? Is according to the size. Now listen this statement. Sir, separation of proteins. Separation of proteins in gel electrophoresis. Separation of what? Proteins. Where in gel electrophoresis is based on number one, charge density. What it is? Charge density. And second one is size. Sir, what is the meaning of charge density? Charge per unit of mass. What is the meaning of charge density? Charge per unit mass. Therefore, separation of proteins based on charge density and size. Separation of DNA fragment based on size. Separation of DNA fragment based on size. Separation of proteins, charge density, and what's our size. Therefore, is this statements okay? Now see this diagram carefully. See this diagram carefully. Right. Sir, in this diagram, right, this is nothing but kit in which we carried out electrophoresis. In this diagram, separation of DNA fragments has taken place. Therefore, DNA fragments are separated by using agarose gel electrophoresis. Is it okay? Clear? Right. By using agarose gel electrophoresis, the DNA fragments gets separated according to their size. According to their size. Is this statement okay? Right. Now, where are these separated DNA fragments? Where DNA fragments are separated? In agarose gel. Therefore, these separated DNA fragments are in the agarose gel. Now, this separated DNA fragments, from this separated DNA fragments, right, desired gene is isolated. What is isolated? Desired gene is isolated. Desired gene is isolated from separated DNA fragments which are present in agarose gel by a process called as elution or elution. Therefore, what is the process name? Elution. What is the meaning of elution? Sir, separated DNA fragments are in agarose gel. From that agarose gel, from the separated DNA fragments, the desired gene is isolated. The desired gene is isolated. What it is called as elution. Therefore, what gets removed? Desired gene. Therefore, removing, right, what? Desired gene. Removing what's a desired gene from where? From agarose gel. From agarose gel. Therefore, removing or isolating the desired gene from agarose gel, that is called as elution process. So, what gets isolated? Desired gene gets isolated. Is this statement clear? Okay. Sir, DNA precipitates when chilled ethanol is added. DNA precipitates when chilled ethanol is added by spooling process. Purified DNA is isolated. Purified DNA is isolated by spooling process. Right? We are cutting the DNA by restriction endonuclease enzyme. DNA fragments are separated by agarose gel electrophoresis. From separated DNA fragments present in agarose gel, the desired gene is isolated. Desired gene is isolated by elution process. The desired gene is isolated by elution process. Therefore, is this statement okay? We completed this flow chart. Therefore, this is a isolation of what? Desired gene. This is isolation of desired gene. Listen here. Note. Number 1, if DNA is circular, it means I am taking which DNA? Circular DNA. If DNA is what, sir? Circular, then number of fragments are formed. When fragments are formed? When we cut it by restriction endonuclease enzyme. Okay, right. Therefore, number of fragments formed is equals to number of palindromic sequence. Number of palindromic sequences. Therefore, number of fragments formed is equals to number of palindromic sequences. Number of palindromic sequences. Number of palindromic sequences on which restriction enzymes acts. On which what acts? Restriction enzymes acts. Is it okay? Right. Number 2, if DNA is linear, if we are taking which DNA, sir? Linear DNA. If we take a linear DNA, in such cases, number of fragments formed. Number of fragments formed. When fragments are forming? When you treat the linear DNA by restriction endonuclease enzyme. Number of fragments formed is equals to what, sir? Number of palindromic sequences. Number of what, sir? Palindromic sequences on which what? Restriction enzymes acts plus 1. Is it okay? Therefore, if DNA is circular, then you cut. If you cut that circular DNA by restriction endonuclease enzyme, then how many fragments are formed, sir? How many palindromic sequences are there? That many number of fragments are formed. Sir, if DNA is linear, sir, right, we are cutting that linear DNA by restriction endonuclease enzyme, sir, then how many fragments are formed? The number of fragments formed is equals to number of palindromic sequences, right, plus 1. Is this statement okay? Right? We are discussing steps involved in genetic engineering process. The first step is isolation of desired gene. Okay, now go for the second step. Insertion of desired gene into a vector. Insertion of what? Desired gene into a vector. Sir, in first step we isolated a desired gene. In first step we isolated a desired gene. Now how many desired genes we are having? One desired gene. We want many copies of desired gene. Sir, having one desired gene, right? Having one desired gene. we want many copies of desired gene. What we want? Many copies of a desired gene. Sir, to get many copies of desired gene, the desired gene should replicate. Desired gene should replicate. What should replicate, sir? The desired gene should replicate. The desired gene should replicate. Sir, for replication, a gene requires origin of replication. For replication, it means to replicate, the gene requires what? Origin of replication. But a desired gene is not having that origin of replication. Is a desired gene having origin of replication? No. That's why we are placing the desired gene in a vector which contains origin of replication. Is this statement clear? Is it okay? Right. Sir, we are having one desired gene, sir. We are having a desired gene, sir. We want many copies of desired gene. To produce many copies of desired gene, the desired gene should replicate. But replication requires replication. But replication what? Requires what it requires? But replication requires origin of replication. Origin of replication. But replication requires origin of replication, which is absent. For a desired gene. Therefore, that desired gene, is it having origin of replication? That desired gene is not having that origin of replication. So, what we are doing? So, desired gene is placed where? Is placed in a vector. Why we are placing it in a vector? Because vector contains origin of replication. So desired gene is placed in a vector which contains origin of replication. Is this statement okay? Sir, having only one desired gene, we want many desired genes. To produce many desired genes, the desired gene should replicate. The desired gene should replicate. But replication requires origin of replication. But desired gene is not having that origin of replication. So we are placing the desired gene in a vector which contains what, sir? Origin of replication. Now you tell me, vector is with origin of replication, yes or no? Yes. Vector is with origin of replication. So vector can replicate. What can replicate? Vector can replicate. But in that vector, what we placed? Desired gene. It means along with vector, now the desired gene also replicates. If vector replicates, the desired gene replicates. If desired gene replicates, we are getting many copies of desired gene. It means what is cloned? The desired gene is cloned. So can we call this as gene cloning process? Yes. Which process? Gene cloning process. Gene cloning process. Is it okay? Right? So desired gene is placed in a vector which contains origin of replication. So what happens, sir? Along with vector, desired gene also replicates. Sir, if desired gene replicates, what happens? Many copies of desired gene produced. Many copies of desired gene produced. Sir, producing many copies of a single gene. What is that process called as a gene cloning process? What is that process called as? Gene cloning process. Because the gene was cloned. Which gene? Desired gene. How? By placing it inside the vector. Why we placed it inside the vector? Because the desired gene does not contain origin of replication. Vector contains origin of replication. Why we want origin of replication? We want many copies. To produce many copies, the gene has to replicate. But replication requires origin of replication, which does not present in a desired gene. So we are placing the desired gene in a vector. Is this statement clear? Is it okay? Right? What is the meaning of a vector? Vector. Sir, vector means nothing but carrier. Vector means nothing but what, sir? Carrier. Now listen the statement. A DNA molecule which carries a foreign DNA fragment, where into a host cell. Into which cell, sir? Host cell. See this statement carefully. Sir, vector means a DNA molecule. What this DNA molecule is doing? It is carrying. What it is carrying? A foreign DNA fragment it is carrying. It is carrying that foreign DNA fragment into what, sir? Host cell. Is it okay? Right. Then what is the meaning of cloning vector, sir? What is the meaning of cloning vector? Name itself indicates. Vector which allows gene cloning. Such vectors are called as cloning vectors. Therefore, vector, what it allows? Vector which allows what? Gene cloning. Vector which is allowing gene cloning. Such vectors are called as what, sir? Cloning vectors. Is it okay? Right. Commonly used cloning vectors. Commonly used cloning vectors. Sir, which vectors are commonly used, sir? Number one, first one, plasmids. Number two, bacteriophage. Number three, plasmid. What it is, sir? Plasmid. Number four, bacterial artificial chromosomes. And last one is yeast artificial chromosomes. Bacterial artificial chromosomes, yeast artificial chromosomes. Sir, plasmids, bacteriophage, plasmid, bacterial artificial chromosomes, yeast artificial chromosomes. These are commonly used cloning vectors. Is this statement okay? Sir, cloning vector means what, sir? It allows cloning. Vector which allows cloning. Sir, when cloning takes place? After inserting the desired gene. After inserting what? Desired gene. Sir, to what size the desired gene should be to insert it inside a vector? For inserting the desired gene into a vector, what should be the size of the desired gene? Right? What should be the size of the desired gene? Sir, what are all these vectors, right? Size of insert. Size of insert means nothing but size of the gene what we are inserting inside the vector. It means the size of the desired gene. Okay, in kb. Kb means kilobase. Okay, right. Sir, plasmid, the insert size should be 0.5 to 8 kb. Sir, bacteriophage, what it is? Bacteriophage, right? The insert size 9 to 23 kb. Sir, plasmid, how much, sir? 30 to 45 kb. Insert size should be 30 to 45 kb. Bacterial artificial chromosome, insert size is 50 to 300 kb. yeast artificial chromosome. Insert size is 1000 to 2500 kb. Is these statements okay? Any doubts in these statements? Therefore, sir, what are all these? All these are nothing but the size of the desired fragment that is to be inserted in a vector. It means, right, up to what size we can insert a desired gene, right, in a plasmids are 0.5 to 8 kb. Is it okay? 9 to 23 kb, right, in bacteriophage, 30 to 45 kb in a plasmid, 50 to 300 kb in bacterial artificial chromosome, 1000 to 2500 kb in yeast artificial chromosome. Yeast artificial chromosome. Now you tell me, sir, which vector is used if the insert size is very large? Yeast artificial chromosome, because your insert size is 1000 to 2500 kb. Is this statement okay? Right. Therefore, for cloning large sized what? DNA fragments. What is used? Yeast artificial chromosomes are used. Therefore, if insert size is large, to clone that right desired gene, we are using what, sir? Yeast artificial chromosomes. Is it okay? Right. Go for the first one. What it is, sir? Plasmids. Plasmids. Now listen. Where these plasmids are present? Present in almost all bacterial species and even in some yeast cells. Present in almost all bacterial species and some what? Yeast cells. Is it okay? Right. In first year, we read some statements concerned to plasmids. Sir, what are plasmids, sir? Plasmids are extra chromosomal. Yes or no? Extra chromosomal, naked, circular, self-replicating, double-stranded DNA molecules. Each and every word in this definition is important. They are extra chromosomal, naked, circular, self-replicating. They can replicate on their own. Double-stranded DNA molecules. Is it okay? Sir, plasmids are inheritable. Plasmids are inheritable. Sir, take a bacterial cell. Which cell, sir? Bacterial cell. For example, this is a bacterial cell, sir. In this bacterial cell, this is the genome. This is what, sir? Genome. Whereas this is plasmid, sir. Compare the size of these two. Whose size is small? Plasmid. Sir, plasmid is DNA, genome is also having DNA, yes or no? DNA segments called as what? Genes. It means plasmid is having genes as well as genome is also having genes, yes or no? Right, but plasmid contains few number of genes. That genes controls a variety of biological functions, controlling a variety of biological functions such as unique phenotype characters, What are the characters we'll discuss now? Is it okay? Right. Therefore, sir, plasmid, we are discussing about what, sir? Plasmid. Plasmid contains few genes compared to what? Bacterial genome. Compared to bacterial genome, it is having few number of genes, right? Present in plasmid. genes present in what? Plasmid. What they do? Controls a variety of biological functions. Controls a variety of what's are biological functions, right? They controls unique phenotype characters. Controls unique phenotype, external appearance, right? Phenotype means phenotypic characters, unique phenotypic characters, right? Sir, genes present in plasmid gives resistance. Gives resistance against antibiotics and drugs. This statement clearly indicates that bacteria is getting resistance against antibiotics due to the presence of some type of genes present in plasmid. Therefore, genes which are present in plasmids giving resistance for a bacterial cell against the antibiotics. Therefore, genes present in plasmid gives resistance against what's are antibiotics and drugs. Genes present in plasmid produces toxins and produces what's are toxins and enzymes. Produces what? Toxins and enzymes. Produces toxins and enzymes. Is it okay? Right? Now go for copy number. What is the meaning of copy number? See here, take a bacterial cell. Take a bacterial cell. In this bacterial cell, I am taking two type of plasmids. I am taking two type of plasmids. One is A plasmid and second one is a B plasmid. What are the two type of plasmids? A plasmid and B plasmids. First you tell me, how many copies of A plasmid is present in this bacterial cell? 2. So what is the copy number of A plasmid? 2. How many plasmids, how many B type of plasmids are present in this bacterial cell? 4. So what is the copy number of B plasmid? 4. Therefore, copy number means number of copies of A plasmid present in a bacterial cell. It means if you take one type of plasmid, how many copies of that plasmid are present in a bacterial cell? That becomes what? Copy number. What it becomes, sir? Copy number. Therefore, copy number means what, sir? Number of copies of a plasmid. Number of copies of a plasmid present in a bacterial cell. Present in a bacterial cell. Sir, what is the copy number of a plasmid, sir? What is the copy number of plasmid? Copy number of plasmid. Sir, copy number of plasmid may be 1 or 2 or 15 to 100 or more than 100. More than what, sir? 100. More than what, sir? 100. Is it okay? So plasmids have high copy number. Who is having high copy number? Plasmids. Plasmids have what? High copy number. Plasmids have high copy number. Is it okay? Right? Thank you.